Hello. Well, thank you for having me here, and I want to thank Sangeeta and the rest of the team for inviting me to give a presentation today. I think I spoke here last time in 2017 when we just published our science paper, so it's been a while. But anyway, today I'll give you an overview about some of the drug delivery technology that we have, and I'll give you examples. A lot of them are in the vaccine space. There's some cancer work that we do as well, but they're really platforms that enable different API delivery under various kinetics. So if you have any insight or interesting applications, please let me know. So this is just sort of an overview of the type of work we do upstairs in my subgroup in the Greater Langer Lab. So um, all of them are polymer-based systems for delivering primarily biologics, but we also work with some small molecules as well. And we have an overreaching goal of addressing some of the really big problems in public health. And that's why vaccines and actually the area of nutrition is very important to a lot of our projects. But today I'm going to talk a little bit about sort of two platforms that we have for drug delivery. On the left there is our long-acting vaccine platform that we use for basically making self-boosting or long-acting vaccines. And again, we've also used it in some immunotherapy, cancer immunotherapy applications. And then I'll talk a bit more about some of the vaccine work we do with microneedles as well that's relevant to mRNA vaccines and maybe has other applications in the cancer space. So I don't think I need to tell anybody why it's important to think about vaccines and how important they can be in cases where we have a pandemic situation globally. But what I really wanted to highlight in this particular graph, this is from last week from WHO, is really the number of vaccine doses over that's 13 billion have been administered globally, to really kind of give you a sense of scale of how many doses and how many applications are needed of this particular technology. And then to highlight sort of this aspect, and for those of you that were here at the Image Awards, we had an image that's outside that's sort of focused on some of these aspects of sort of vaccine equity and getting the vaccines and the drugs to everyone. And as you can see, there's a clear area that's underrepresented in this. And so part of our work is funded by the Gates Foundation. And it's also personally very dear to me to really try to address some of this inequity that exists in the world today. So with that, I'll sort of jump into the science. And again, the work that motivated all this was pre-pandemic, and it was really focused on these developing world applications where many children die because they're under vaccinated, and they're actually able to get one shot, in some cases two, but the data shows that because they don't get the full course of vaccinations needed for particular childhood diseases like polio, for example, they end up being unprotected against the pathogen and can have deadly consequences. And so our idea was very simple. Can we take these multiple shots of vaccines, and can we actually incorporate multiple vaccines together into one shot and give it to administer it to kids and potentially even adults? And obviously, in the case of a pandemic, which we recently experienced, this could be highly useful because we could have better patient compliance. As we've seen the data, as you require more shots, we have less and less people getting vaccinated with each shot. There's also potential to really look at the kinetics and if there's a way to get better and more durable responses because we can offer multiple shots from that single injection. And then, of course, in the case of pandemic circumstances, one would imagine if you had more durable, stronger responses, that you have less probability of variants occurring in the population. And so really the fundamental part, which in some sense is very simple, that allowed us to create this system, it really goes down to materials and controlled drug delivery. So in general, the last 30, 40 years, a lot of the biologics, proteins, and this work was pioneered by Bob Langer and others, were delivered from microspheres, so polymers, which have the proteins distributed throughout. For example, you could have other type of vaccines, but for example, proteins can be distributed throughout the microparticle. And this allows you, once this is injected in the body, or rather hydrated, it allows you to create continuous kinetics, and you can actually play around with the formulation, and you could get a pulsatile type of kinetics, which, as you know, are very important for vaccines. You want to have the vaccination, then nothing for a certain period of time, and then another pulse, if you will. And so our hypothesis was super simple. If we could create a core shell structure, then we'd be able to really control the kinetics as much as we want by just changing the shell of the microparticle. So just think about an egg and not having the yolk come out until the shell is cracked, rather than diffusing through it. That was the concept, and we had a really hard time to actually make this happen at the scale that's needed for injection. So really small particles, smaller than a grain of sand. And so we failed miserably, and we collaborated with others, and we tried all the tricks and really couldn't get it to work. And I had a postdoc, Tan Nguyen, and he's now a professor in mechanical engineering at UConn. And he said, well, I think there's a way to do this if we do some things the way they do it in microelectronics, because they make all these structures at a fine scale. And so it took a lot of effort, but we were finally able to create this wonderful method. And the key part to it was we wanted to use materials which had a proven record of safety, like PLGAs, which are used as resorbable sutures, and could be used in children and infants and essentially anybody. And so we were able to develop a method that allows you to make these tiny little particles. They ended up being cubed rather than spherical. And you can fill them with anything you wish to some extent. And these are just some quick videos I like to show because it's easier to visualize. But we created these material particles. So these are bases that are made out of these materials, and they can be made out of any actual thermoplastic. And then we have a way to fill them in a high throughput way with piezoelectric mechanical arms, just like you would find in an inkjet printer. And then we have the lids that come in and seal them. So as you can imagine, it's quite versatile. Here we have 4. We make these on slides of 1,500 particles per slide. And we're now scaling that to even larger. And this is just another image, an actual optical image of these particles being filled by the robot upstairs. And you can see that's a blue dye. So you can imagine you can really fill them with any components that you like. And then just to show you, the important part is the sealing. So if you focus, it might be really hard, but on this particle, there's a diffraction pattern on the wall of this particle, and we're able to optically follow this. You can see it moving now across the screen. And as the particle is sealed, that diffraction pattern, which is just the air and material interface, will disappear, and you can have an optical algorithm or AI program that would tell you if the particles are sealed or not. So it really has the ability to be scaled in a throughput manner. Here's just some more beautiful images of these particles. Again, we make these in large arrays. I'm used to doing Zoom talks, so I should have brought some samples with me, but I didn't. But next time I will. But anyway, you can kind of see what they look like. And this really is the important aspect here. What I'm showing is that by changing the chemistry of the materials that we make the particles from, we can change when these release in vivo. So this graph here on the left shows cumulative release from these particles in vivo over time. And you can see that we can control when these particles release on the order of days to weeks, months, and even out to 6 months. In fact, we can have these particles stable for years. The challenge, of course, is stabilizing the drug that you want to deliver, especially when you're talking about biologics and of course, mRNA. But with these release kinetics, we then went and did studies with several vaccines. This is an example of the Sabin inactivated polio vaccine, which you're required to have 3 injections as a child of this particular vaccine. And what we did here is we develop these particles, we had to do until mRNA vaccines. This was considered as one of the most thermoliable vaccines out there. And we spent a lot of time trying to stabilize it for over this 2-month period in the body so that when it does release, it gives you a strong enough response. And so what you see here in red is, or maybe I'll start with the black, is the positive control. So this is 3 shots of the same vaccine. This vaccine has 3 different serotypes, so those are the 3 different graphs. And then our blue actually shows are what we call unbuffered particles. So these particles are just made from the polymer, and you can see they didn't do so well. And so what we realized is that this polymer degrades into acidic byproducts. And so just before the release point, there's a dip in pH. And so we started making ones that sort of decrease the amount of acidic byproducts that are experienced by the cargo inside the particle. And that allowed us to have these very nice single injection neutralizing, these are neutralizing titers that are actually done by the CDC for polio from using our particles. So a single injection of the red versus the three in black. And these are now in monkeys, non-human primates, and we actually have some of the data trickling in, and it looks very similar to this. So we're really excited about that work. Similarly, again, I want to emphasize the platform of this technology. So we were able to, again, put different things inside. So here's an example where we put a hepatitis B vaccine. The polio vaccine is a nanoparticle. It's an inactivated, formaldehyde inactivated vaccine. It's about 27 nanometers in diameter. This is the hepatitis B vaccine, which is a recombinant protein. And this was done with a CpG adjuvant together, and you can see the data for the mouse studies are really great. Again, we have a single, this is only 2 presentations, so 0 and 1 month. And you can see from a single injection, we get the same responses as with multiple injections. We also had really nice stability at room temperature with this vaccine as well. This is also now in non-human primates, and we certainly have responders, so we're very excited about this work as well. And then another example is, this is what we call the T. solium vaccine. T. solium is a tapeworm disease that's prevalent in, it's one of the tropical neglected diseases that has actually potential to be eradicated. The issue is you eat uncooked pork and you get this, and it actually leads to, it gets onto your brain and it leads to epilepsy. So it's a quite rough disease or harsh disease. And again, we're looking for a 0-1 month time point in the release kinetics to deliver this vaccine in 1 shot. And again, we have very nice data in mice, and we have very nice storage data. This uses a saponin-based adjuvant as well with it. And again, this is now the idea here is that you vaccinate the pigs rather than the humans because they're the ones that carry it. And this is now in pigs, so the target animal at Tufts University Veterinary School. And again, we're seeing responses, so we're very excited. So with that, I'll just kind of summarize the work in vaccines here. So this is sort of our self-boosting long-acting vaccine program. I talked about the first row, and a lot of people ask, well, how many particles do you need to vaccinate? Because, you know, how much can you put in there? So for a monkey study, which is about half a human dose, we use 280 particles for the 3 serotypes of polio and the 2 extra, so the 1 month and the 2-month time point. So it's actually not a lot of particles, and they are really small. They're still relatively large, so you have to use like a larger needle than what they currently use clinically, but we're working on miniaturizing them. And again, actually the pig vaccine requires a bit more particle. It's a pretty high dose. And of course, we're also working with influenza. And now we're very much working with mRNA-based vaccines. And that's some of the image awards. Have some beautiful pictures of some of that work that's still in progress because of the challenges associated with stability, as you know. I'm running out of time, so I'm just going to quickly go that this is probably most important, most interesting to all of you. We've also used this work to deliver STING agonists in different cancer models. This is published as well, so but I'm happy to talk more about it, but I'll go through it very quick. So we, again, we were just comparing, a lot of our initial studies have been really showing that technology works, so we're just comparing it to sort of the equivalent multi-injection therapeutic. And now we are actually looking to study more how we can control kinetics beyond what's been studied clinically before, to see if we can further modulate or further excite the immune system to do things that we couldn't do before. But anyway, in this example, this is a melanoma mouse model. And so after the tumor was induced, we then had a surgical removal, and we either did multiple injections of a STING agonist, or we did a single injection of our particles that were left behind. And both for the post-surgical removal, we had no statistical difference between the multiple administration. And again, similarly, after a 2-month re-challenge, we had the same, no statistical difference between multiple shots versus ours. We then also looked at a metastatic 4T1 model with these particles, and again, similarly, so this was a breast metastatic model and we were looking at the lungs. And again, we were able to show that there's no statistical difference between the metastatic foci between our the three classical injections versus just our particles. And in fact, we even saw a better outcome when it came to the area per the metastatic area per lung area as a percentage. So again, showing that it's and as you can imagine, these are easy to inject in some of these intratumorally, especially post-surgically, and of course they greatly decrease the number of injections that a patient is required. Yeah. And while we didn't encapsulate, we also looked at in combination therapy. We didn't encapsulate the ICB here, the PD-1 in this case, but when we gave it in conjunction with our particles, again, we show a decreased tumor volume in the primary treated tumor, and we also saw an effect on a distant tumor that was injected at the same time or inoculated at the same time. And in this case, this was, again, the melanoma model. We also showed a pancreatic therapeutic efficacy of these STING microparticles in a pancreatic model. And again, in this case, we weren't able to compare it to a multiple injection system. But again, we showed really decrease in tumor volume. Again, we're using these particles. So with that, I think I've highlighted the sort of applications of these technologies, both for something like vaccines and something like cancer immunotherapy. Again, they're really a platform, and so they allow us to really mix and match the particles and really adjust the kinetics as needed. So one can imagine maybe some future studies where instead of giving STING agonists 3 times over a 2-week period, maybe we give it 8, or maybe we give it 2, or we give it in combination with some other type of kinetics in a certain period of time. So maybe briefly in 3 minutes, I'll go over our micronutrient technology. What? Okay, but I don't want to take, okay. Okay, so again, I wanted to kind of highlight this vaccine equity aspect. And what we learned in this particular pandemic is that there were parts of the world that not only didn't have the vaccines as early as we all did, but also weren't able to even accept vaccines at some point because of the storage requirements and the logistics of just handling these type of vaccines. And so we actually started, so our idea was, is there a way to do decentralized vaccine manufacturing. And actually, this work started before the pandemic, and some program officers from BARDA gave me a call, and they said, well, we really are looking to do some sort of on-demand vaccine manufacturing because we don't want to stockpile a vaccine in case there is a pandemic. And we want to be able to sort of ship them to a place and just have it on demand there. For example, they used Ebola as an example. If there's an outbreak of Ebola, you could ship this there, vaccinate the local community. So what we came up with is this printer, if you will, that could really, it's not that large. It could fit on top of a table. It's upstairs on our lab bench. But the ink for this printer would contain the vaccine and the materials needed to make microneedles. So for those of you that don't know what microneedles are, they're again, I'm really bad with this bringing samples to talks, but they can be like a centimeter square. You can have about 100 needles there. And in our case, we make them dissolvable, so the ones that don't leave any sharps. And you could put in the tips of them, you can put a vaccine or other things, and you can apply them with your finger to your skin. They'll puncture through, and the tips will stay dissolved in there and stay behind after about 2 to 5 minutes. And so it's a great technology. It doesn't involve any sharps. You don't need to have a healthcare worker that's trained in administering needles and working with syringes and all of that. And there you have some images, SEM images of the microneedle. So we were working on this project during the pandemic, and so we decided to shift from Ebola to mRNA vaccines. And to maybe quickly summarize the work there, we did a lot of sort of mechanical engineering in this project, where we were able to develop a system to make microneedles. You need to pull them into a mold, and the tips need to be super sharp in order for it to work. And so you really need a centrifuge. That's usually how it's done. But unfortunately, we didn't have that ability in this standalone device that just sort of needed to have one plug. And so we developed a vacuum through method, and you can see here in the middle, you can see those needles being filled. So the vacuum goes below the molds and is able to fill these sort of sheets. You can see on the right, these are the sheets that come out of the printer, if you will, that contain the different patches that would contain the vaccine. And to sort of emphasize, the ink was a combination of mRNA LNPs, in this case, encoding for luciferase and then later for COVID, and then also polymers like PVA and PVP that are commonly used to make microneedles. But the ink was specially formulated for stability of the mRNA. And there's a lot of mechanical aspect to this. Drying and forming these is very important. And so I won't go into all of these details. And of course, the high throughput, again, what we're seeing here, this is not something to sort of replace making 13 billion doses of vaccine. This is something one could take to, for example, a refugee camp or an emergency situation like what's going on in Turkey after the earthquake, or an outbreak, a small outbreak of Ebola or something similar, where you could bring it to one of these areas or even a military base, and you could make vaccines on demand and vaccinate the people there fairly easily to prevent the spread of disease in these sort of tight quarters. So we're talking more tens of thousands, 100,000. Could also be used for sort of clinical development in small labs, especially in areas in the world that don't have access to manufacturing and big pharma companies they can collaborate with. So this slide actually summarizes the work very well. The big aspect was, how do we fill the vaccine? How do we stabilize it in the liquid? And then how do we fill it so it remains stable once it's in the tip of the nanoparticles? And that took quite a bit of work and sort of screening of different formulations. And really, there was sort of two opposing forces. One was to really create these microneedles that can penetrate the skin and are stable from an administration point of view, but then also to stabilize the LNPs and of course the mRNA inside the LNPs. And so we were able to reach sort of an ideal situation where we sort of compromised a little bit on each, but we had good, very high bioactivity and very good mechanical strength for administration. And a lot of detailed science and fundamental work went into developing these things. I won't go into it for time. And then we ended up using mRNA encoding for SARS-CoV-2. We did this very early on, so our sequence is a bit different than what's Moderna and Pfizer sequence or the commercial ones, but nonetheless, it's still a good sequence. And this is our LNP composition as well. So slightly different than perhaps the commercial ones. And this sort of just summarizes some of the way that we factored in mRNA stability and the microneedle, the printing part, and then also the stiffness of the final microneedles. Again, I won't go into the really vacuum step on how we created these needles in this printer are very important. So the engineering aspect was very important, and to ensure we're not wasting any of the precious cargo that's only in the tips, which is shown here. That also increased our loading and efficacy of delivering the mRNA vaccine. And then this, I'll just end with a couple slides here, which highlight, so what you see here in black, and in black and red are the red is the microparticles, so the solid formulation of the LNPs inside the microneedles, and black is the soluble version. You can see over 6 months storage, they both do pretty well, at least this formulation at 4 degrees Celsius. But when we keep it at room temperature, 25 degrees, which is our room temperature here, you can see that the solid formulation in the microneedles outperforms by far the formulation that was stored in a liquid form at the room temperature. So this was really great data. This was with luciferase. And then we also looked at it at really high temperatures, so 37 degrees C, and we had very nice, again, stability under these conditions. And then finally, we incorporated the RBD sequence for mRNA, for COVID surface protein in the mRNA. And in this case, again, we're just comparing how our patches compare to a regular soluble injection into the muscle. And you can see we had very nice responses that are completely comparable to those of the regular administration. We also looked at various variants, again, against COVID, and we had very great data as well, consistent between the patch and the actual needle injections. And we also looked at 3-month storage at room temperature for the mRNA vaccine. And again, we had fantastic data there with looking at the titers after administration. So with that, I will end, and I want to thank so much for your attention. But most importantly, I want to thank for the incredible team that works upstairs with me. And of course, I want to thank Bob for just being a fantastic mentor and collaborator, and other Dan Anderson and Dan Buruk over at Harvard, who does a lot of work with us on the vaccine. And of course, our funding, NIH, BARDA, and the Gates Foundation. And finally, I just want to put a close up. There's no names here, but I'm sure you recognize a lot of these people that you see in the elevator. So with that, thank you, and I'll take any questions.